What we did, we took human stem cells that were derived years ago from donated early embryos, or what's called iPS cells, cells that are taken from the skin and turned back into a stem cell state. These are very established technologies. And what we do this time is we put them in a special media that takes them to really, really early stages, what we call naive. So we have to use these human naive stem cells. And then we put each aggregates of 120 cells. And again, in about 1% of the aggregates, we can see that cells start differentiating correctly, migrating and sorting themselves into the correct structure. And the farthest we could get is day 14 in human embryo development. The human embryo makes all its organs basically by week 5 of pregnancy. And we really don't know a lot. It's a black box of how organ formation happens. And it's almost impossible to study this period because the embryo is very small. There are justified ethical limitations to really access such material. Therefore, the goal is really, can we bypass and create some kind of alternative model from cells, from stem cells, and try to make a model for a human embryo. By making these models, and that are very similar, and we can start with stem cells that are engineered, labeled, knock out a gene, and so forth, so we can really know which hormones and which genes are important for which organ, and from that knowledge, we can try and go back and improve our differentiation protocol. And maybe actually in these embryo models, which is in the end some kind of a differentiation protocols, there might be some cells that are useful actually that you can take and transplant, or take them and expand them and transplant them, or take them and test drugs on them and so forth. So they could be also themselves as a source of cells.
A group of scientists from the US and UK teaming up to create what is being called the world's first synthetic human embryos made without eggs or sperm. Some are calling it a scientific breakthrough. Others say it raises serious ethical and legal questions.
Yeah, it's a terrifying prospect, and when you read about it, it feels a little bit Black Mirror-ish in a way.
Black Mirror is all about, you know, shining a light on, you know, kind of like the darkest part.
Everything that's
the
underbelly.
The underbelly,
Oh, that's, yes.
Yes, the great word.
I am part of the underbelly.
You are?
We were actually just talking about artificial intelligence earlier in the show, and talk of AI is a major part of the episode.
Huge.
Could you see that plot perhaps playing out in real life?
I think it is playing out in real life, which is, you know, it's a spooky thing.
Here's what's going on here. They took one single cell, embryonic stem cell, one single cell, and they're manipulating it to see what the placenta would look like, what the yolk sac would look like. They're not creating actual embryos that you could do something with. There's no precursor of a brain or a heart, and they're not planning on using it for anything other than testing why someone miscarries, or what are genetic abnormalities and can you predict them. But I have a question. I would have a question for the researcher here, same as I have a question about gain-of-function research. Could you do this with computers? Could you do this with artificial intelligence? Could you predict this without using this actual cell and manipulating it?
As The Guardian notes, this discovery also falls beyond the realm of regulation in the UK and in most countries, so it does raise a lot of ethical issues. The embryos don't have a beating heart or any brain matter, but they have enough matter to form a placenta, an egg yolk, and embryo. So, as I said, raising major ethical and legal questions as well.